Good morning, everyone. I'm Zikra, a third-year PhD student here at NII. It is my absolute pleasure to welcome you all to admire, cherish, and celebrate the National Institute of Immunology on its 36th Foundation Day. With all my heart, I welcome the guest of honor, Professor Sylviane Muller from University of Strasbourg, France. I wholeheartedly welcome Director NII, Dr. Debashish Mohanty, and our founding director, Dr. G.P. Talwar. Finally, yet importantly, I welcome all the distinguished guests, staff, scientists, and students of NII to this wonderful occasion. Approximately 4 decades ago, the vision and dream of Professor G.P. Talwar turned this barren landscape into a beautiful institute that we call NII. On 6th of October in 1986, the then Prime Minister, Mr. Rajiv Gandhi, dedicated the institute to the nation. Over 36 years, many great minds of the nation have come together and turned Professor Talwar's vision into a reality. I'm not a poet myself, but a couplet of Majrooh Sultanpuri comes to mind. मैं अकेला ही चला था जानिबे मंजिल मगर लोग साथ आते गए और कारवां बनता गया. It means when I started my journey towards my destination, I was all alone in my efforts. But as I made progress, others came forward and joined me, and we became a force to reckon with. The National Institute of Immunology is a testimony to this couplet. The institute reflects the power of one vision, one dream. It is a testimony to the fact that the journey of many miles begins with a single step. You take that one step, and others shall join you too. In the last 36 years, NII has flourished in cutting-edge research in the field of immunology, infectious diseases, reproductive biology, and structural and chemical biology, as evident from several high-end publications. NII has always strived to connect the excellence in basic fundamental research with the realistic quest of developing innovative applications possible. We hope to continue this tradition and build upon the good work set under the guidance of all the great minds that have led this institute to its glory. So, one such leader that we look up to is the current director, Dr. Debashish Mohanty. Please join me in welcoming Dr. Debashish Mohanty to the stage. So I request you to please come forward and address the gathering.
Sure. Thank you. And Professor Sylviane Muller, our chief guest for the event, Professor Talwar, our founder director, all guests and dignitaries, my fellow faculty colleagues at NII, our students, staffs of NII. I welcome you all to the 36th Foundation Day ceremony of National Institute of Immunology. This Foundation Day is even more special as we are also celebrating Azadi Ki Amrut Mahotsav, which is a year-long celebration by Government of India to commemorate 75th year of India's independence. So our institute was formally dedicated to the nation by the Prime Minister, the then Prime Minister, on 6th October 1986. Hence, every year we have been celebrating the Foundation Day on 6th October. However, the true birth of NII goes back to years of hard work prior to 86 by our visionary director, founder director, Professor G.P. Talwar. So we are happy to note that like every year, he has been able to join us for this celebration. We really appreciate it. So NII Society was formed in 1981 when Department of Science and Technology of Government of India identified basic and applied immunology as a priority area in recognition of its potential impact for human health. So Professor Talwar was invited to lead this institute as founder director. After operating for initial few years at AIIMS and in other rental complexes, NII started functioning in this campus from our research RSH, what we call RSH, our research scholar home, in 1983-84 around that, and until this magnificent main building came up in 1986, and that is when it was dedicated to the nation, and we have been celebrating our Foundation Day as 6th October. So in the first decade of its existence, that is almost up to 91-92, NII made major contributions in the area of vaccine development, reproductive immunology, most notable being the successful development of leprosy vaccine based on Mycobacterium indicus pranae, which was discovered in NII by Professor Talwar and his group. And it may be pertinent to mention that utility of MIP, Mycobacterium indicus pranae, as an immune modulator has also subsequently led to exploration of its efficacy as in other therapeutics for sepsis and also recently for COVID-19. So it is obviously a great achievement for us. And in the next 1.5 decade, that is up to 91 to 2007, NII further strengthened its position as a premier research institute in India and made innovative contributions in basic immunology, infectious disease research, virology, chemical biology, structural and computational biology, as evident from several high-impact publications. During the last 15 years, NII has acquired cutting-edge modern infrastructure for research and has made notable contributions in cancer biology, systems immunology, aging research, glycobiology, and structure-based drug discovery. During this period, NII also strengthened its PhD program to attract around 25 bright and talented research scholars every year through a highly competitive national level entrance examination. And apart from our publications and patents, the success of NII is also evident from the fact that 8 of our faculties members have been recognized by Santi Swarup Bhatnagar Prize, the most prestigious award in Indian science. NII faculties have a significantly high share in fellowships of leading science academies in the country. 9 faculty members from NII have been selected to take up leadership positions of major research institutes in India, and that includes Dr. Rajesh Gokhale, who was our faculty till he joined now as the Secretary of the Department of Biotechnology of the Government of India. And I must acknowledge the contributions of our founder director, Professor Talwar, and the role of subsequent directors, Dr. Sandeep Basu, Professor Surolia, Dr. Chandrima Saha, and Dr. Amulya Panda for our achievements in the last 4 decades. It is needless to mention that the success of NII is not only because of our faculties and research scholars, but also our extremely talented technical, administrative, dedicated technical, administrative, and support staff. I must mention that without the continuous support of Dr. Rajesh Gokhale, the current Secretary of DBT, and all past secretaries of DBT, this would not have been possible. And we put on record our appreciation of the continuous support DB Department of Biotechnology has provided us. So while it is natural that we take pride in our achievements, it is also a time for us to consolidate our efforts to get ourselves ready for future challenges. The Foundation Day after the pandemic is the right time for introspection. The successful development of vaccine within a short span of time has made common man aware of the power of immunology. It is natural that NII will be expected to play a still bigger role in vaccine research and disease biology. And it will be necessary for us to integrate the laboratory research, which is our current strength, with translational health science and public health. In recognition of this challenge, during the tenure of my senior colleague, Dr. Pushkar Sharma, who was officiating director, we have initiated the efforts to start network programs on human immunology involving NII as well as Clinical Research Institute, and such collaborative programs in few other areas are also being planned. And we always look forward for the Foundation Day talk, which is very special to NII fraternity. And today's Foundation Day talk is by very eminent immunologist, Professor Sylviane Muller from Strasbourg University, and she will provide us valuable insights into how mechanistic insights obtained from laboratory research can be converted into therapies for translational applications. I must thank Professor Muller for accepting our invitation. I must also thank Professor Srini Kaveri from CNRS and Professor Nitin Seth from CEFIPRA for facilitating the visit of Professor Muller to to NII because she has been invited as CEFIPRA to give several talks in India in many research institutes. And NII in the past has had active collaboration with leading research institutes in France right from the time of Professor Talwar. And Professor Talwar, he has been recognized as the highest award, civilian award for France. And we have another, Professor Srini Kaveri is another such awardee probably in this audience. It is a matter of pride for us. So this afternoon, we are also planning to have an interactive session with Professor Muller and colleagues from CEFIPRA and CNRS, which we can have further discussions how to enhance the scope of our future collaboration. And without taking much of a time, I looking forward to a memorable Foundation Day celebration, and thank you all for joining. Thank you.
Thank you, sir. I now call upon stage Dr. Veena Patil to introduce Professor Muller to the audience.
Hi everyone. It's indeed an honor and a great pleasure to be introducing Professor Sylviane Muller. I hope I'm pronouncing it correct. So Professor Muller is a professor at the Institute of Advanced Studies of Strasbourg University, where she holds the chair in the therapeutic immunology. Professor Muller obtained her Doctor of Science from University of Strasbourg, France, and postdoctoral training from Max Planck Institute of Immunobiology in Freiburg, Germany. So Professor Muller has held many prestigious positions. From 2001 to 2017, she has served as a chair and director of immunopathology and therapeutic chemistry unit of French National Center for Scientific Research, that is CNRS. And from 2016 to 17, she has also served as a former director of CNRS, the Institute for Molecular and Cellular Biology in Strasbourg. From 2011, for almost about a decade, Professor Muller directed the Medalist Drug Discovery Center for Cancer and Inflammation, a laboratory of excellence funded by the Excellence Initiative of University of Strasbourg. Medalist is succeeded as of 1st of January 2021 by the new Strasbourg Drug Discovery and Development Institute, of which Professor Muller is the first director. She has also contributed as a member of the CARE Committee for COVID-19 in 2020. She's a member of editorial board and ad hoc reviewer in several scientific journals, member of several international scientific societies, expert in national and international selection board and committees, member of several scientific, academic, and private advisory boards. Professor Muller is also a fellow of European Academy of Science in 2020 and a member of Academy of Europea in 2020. She's a co-founder of Neosystem, which is today called as Polypeptide France, and Immune Pharma France, the two companies that she has founded. Considering her contribution to the field of science, or especially in the immunology and with autoimmunity as a focus, she has won many prestigious awards. CNRS awarded Professor Muller a silver medal in 2010 and an innovation medal in 2015, a prestigious award that recognizes individuals whose contribution, outstanding research, leads to significant technological, therapeutic, and societal innovations. Professor Muller has received Leon Vellus Prize from French Academy of Science in 2016 and was a finalist of 2017 European Innovation Inventor Award in the category of research. In 2017, Professor Muller was awarded a Prix d'Honneur and an innovation trophy by French Arthritis Foundation. Professor Muller was elected as a member of Chemical Sciences section of the Academia Europea and the Chemistry Division of the European Academy of Science in 2020. Professor Muller was promoted to the rank of officer of the French National Order of Legion of Honor in 2021 after having been named as a knight and officer of the National Order of Merit in 2016. She's also on the board of USIAS, where she actually positions the secretary of the board and supporting the director in representation and the strategic decision making. Coming to her research, I'm sure many of you already know, she is world-renowned immunologist, has contributed significantly in the field of autoimmunity. As everybody here knows, that autoimmunity is treated by immune suppressants, but she has an innovative technology where she has used synthetic peptides to treat the autoimmunity. So her work has been focused on understanding the molecular and cellular pathways involved in the autoreactive lymphocytes activation, the events that lead to the cell death or living phenomenon that are central to the auto-inflammatory diseases. Using synthetic peptides as a tool, Professor Muller has characterized the autoantibodies and autoreactive T lymphocytes, especially in human lupus, and the design of the innovative vaccines to the pathogens. Especially combining her fundamental knowledge of lupus with a long-lasting experience in peptide immunochemistry, Professor Muller and her team were able to develop a very novel strategy based on the synthetic peptides designed to modulate the aberrant immune response and restore immune system function in mouse models as well as in patients with lupus. One of the peptides is currently evaluated worldwide in phase 3 clinical trials. She has, to her credit, many patents, over 30 patents, and over 400 publications that are published in highly reputed, high-impact journals, including Cell, Nature Medicine, Autophagy, Trends in Immunology, Trends in Pharmacology. With this, without taking much of your time, so I'm sure all of you are more eager to listen to her in terms of how she went into her journey from understanding the disease to finding a therapeutic value using the novel peptide tools. So I welcome Dr. Muller to deliver her talk on mechanisms-guided novel therapies for treating SLP, that is lupus, and the other auto-inflammatory diseases. Professor Muller.
I'm really impressed by this introduction, and really I thank you so much for being here, and I'm very glad to be in India, only the second time in my life, and to discover a part of the world that I don't know very well, and I discover again with a high pleasure. So thank you for attending, and I will explain what you are doing and what is a novelty, and you will see how you invented something, a concept, and year after year we continue on our line to give something to the patients, because it is our only purpose to provide a drug to treat so many patients with inflammatory disease in the world. So just a few words about what is autoimmunity, because I'm here in an institute of immunology, but perhaps some people here has no background in autoimmunity. So as you know, the immune system normally recognize foreign antigens and invaders, but sometimes it make mistakes and recognize our own components as foreign, and by definition induce an immune reaction against our own cells and tissues. And this is shown by the production of autoantibodies, so antibodies against our own components, and lymphocytes that will react to eliminate this self. And of course, this in you and me, we all have a small part of autoimmunity, but this is in general harmful. It's no detection any longer, but sometimes in certain circumstances, we have a switch to an autoimmune diseases. So in this case, we have a progression from something benign to something pathogenic. And it is determined by different factors, genetic influence, but also environmental triggers. And this can affect one particular organ or tissue or all the body. So we talk about organ-specific autoimmune disease or systemic autoimmune diseases. But the border between the two categories is not so strict. So we estimate today, of course, it's difficult to have clear figures, but we estimate that between 5 to 8% of the total population in the world has an autoimmune disease. So personally, I work with lupus for a long time now, and it is for several reasons, and also because initially I'm a biochemist, and I was interested by the structure of chromatin, the nucleus in our cells. And patients with lupus produce antibodies against nuclear antigens. So we know that more or less 5 million people have lupus in the world. Most, as you can see, are women, and this is partly due to the fact that the origin of the disease, this particular disease, is, as I said, multigenic and environmental, but also hormonal. So in this disease, which is particularly difficult to diagnose in certain cases, multiple organs can be affected. So you can have skin, as you can see, this rash, this typical rash on the face, joints, kidney, brain, heart, lungs. People have blood clotting problems. And at the level of the immune system, there are more or less everything is affected, with complement deficiencies, modification of cytokine secretion, hypergammaglobulinemia, the production of a large panel of autoantibodies, and the production of immune complex that can deposit in the kidney and block the kidney. So one form is particularly hard. It's a neuropsychiatric lupus. In this case, there is a rate of mortality which is increased by a factor 10. And so people are particularly affected, and it's difficult to diagnose properly. There is also a form of neonatal lupus, and in fact, the fetus or the newborn is not properly affected, but this is due to the passage of antibody from the mother to the fetus. And these antibodies we characterized in our lab some years ago react with a particular antigen present in the fetal heart. And so the baby either there is dead or it can be delivered, but we have to pose a system to help them at the level of the heart, a pacemaker, and we have to treat. Sometimes they still die. So, and for all this scheme, we have no cure at all. So what we have treatments, but they are personalized because you don't treat a person with a kidney disease or with a brain disease, but they are just palliative. We have immunosuppressive drugs like corticoids and others, immunosuppressors, but with no specific effects and with a number of side effects that are deleterious. So today we have to say that thanks to these drugs, anyway, we have a higher life expectancy than before. Because before the appearance of cortisone, for example, we spoke about 50% death in 1 year. So today we have a better issue with 95% survival after 10 years, but the quality of life is quite affected for the reason I said. And today the mortality in lupus patients is 3 times more than in the normal population. And in general, people die from renal disease, cardiovascular disease, and also from infection. And infection is mostly due to the treatment we have to impose. So today we come with another strategy, completely different strategy, not at all suppressive, and which is an immunomodulator. And also that in the pipeline there are more than 70 clinical trials they are currently running. Very few can reach a phase 3 of experiment. So with our product, which is called Lupusor, we are among the few ones in the world in phase 3 stage. So the peptide present in the Lupusor product has been discovered by chance. It was not a prediction before starting. During the screen of a battery of overlapping peptides present in an autoantigen which is recognized by antibodies developed by patients. This protein is called U170K protein, and it's part of this big complex you can see on the right, containing several proteins and an RNA, which is particular for this particle, spliceosomal particle. And only one peptide coming from this, all this panel of peptides were recognized by the T cell from lupus mice. So we were, of course, interested by this peptide, and we changed the composition. We introduced post-translational modifications like phosphorylation, acetylation, etc. And one peptide containing a phosphoserine at position 140 was very well recognized by T cells from lupus mice. This is the one in pink. So a few years later, we discovered that the patients with lupus had cells that recognize the peptide. But in contrast to what we had in the mice, in patients, we had no proliferation of T cells. On the contrary, we had a blockage of their multiplications. No proliferation and no secretion of pro-inflammatory cytokine. On the contrary, production of IL-10, which is a regulatory cytokine. So the cherry on the cake, we discovered that when we treat lupus mice that develop this disease with a peptide, we could increase their survival significantly. This is the curve in green. We can decrease a number of clinical signs, dermatitis, proteinuria, damages at the level of the kidney, and decrease also the production of damaging autoantibodies reacting with double-stranded DNA. So this peptide was patented. The first patent was 100% CNRS, the organism in which I'm a researcher. And then we created a company, a startup called ImmuPharma, and CNRS delivers the license of this patent to the company. So then over the year, and I like to put all the steps because you see that it's so long when you patent something, and then to really inject the peptide to patients in significant lupus clinical trials. So phase 1, phase 2B, phase 2A, phase 2B, and the results were really promising, and we enter in the phase 3 in all over the world. So this phase 3 was very good in Europe, and also in Mauritius, where 80% of patients were from Indian origin. But in the US, it was completely negative. No difference between the placebo and the active group. So today, we repeat this phase in the US with a US company called Avian in the States, and this is running now. So, and we, just as an improvement also, we started now another clinical trial for another application. It's chronic inflammatory debilitating polyneuropathy, CIDP, so it's really now something we extend. So just to make the story short, between the phase 2b and the phase 3, we had no great difference. So I just summarize here the results. For example, for the phase 3 in Europe and in Mauritius, we had 71% responder in the group of patients that received the peptide versus 49% of patients that receive the placebo. So this now is in continuation, and we hope that the phase 3, the second phase 3, will be successful, of course. So this peptide, for those interesting, is quite interesting. We have now the NMR structure, and we know where it is located on the protein, as you see within the cycle. So very well exposed at the surface of the particle. This peptide is beautiful, very kind with us, is highly soluble, non-toxic, non-immunogenic for itself, and very stable. So we are really lucky with this component. So we know how the peptide enter in a mouse. It enter and will locate in the spleen where you have the immune cells. And we know that the peptide, after intravenous injection, will be found in the marginal and mental zone of the follicle in the spleen. So a region very rich in macrophages and B lymphocytes. Also, another student, Christophe Macri, discovered how the peptide enter into cells. So the peptide enter via the clathrin pathway, and after a while, we have the peptide in the endosomes and then in the lysosome. And you will see the lysosome in cells is really a key compartment for the activity of the peptide. Another student identified the partner of the peptide, which is an HSP protein, HSP protein, which has a particularity to be constitutively exposed. By definition, an HSP protein is expressed when you have a shock, when you have a stress. But this member is always exposed, and Nicolas discovered that the level of expression of this HSP protein at the cell surface is still increased in case of inflammation, particularly in case of lupus structure. We know where the peptide bind in the structure, so all that was done during the years. So you see the publication we have made with this aspect. So, and we discovered that the peptide, when it is attached to the protein, hampers its chaperone protein and reduce the interaction of the HSP protein. The old name is HSC70, it's a bit complicated. So the interactome was completely destroyed when the peptide were there. So what could be the impact of this? The impact of this disturbance should be that the chaperone protein cannot protect the molecule that present peptide to T cells. And if it's true, the quality of peptide that are presented in the context of MHC molecule at the surface of antigen-presenting cell should be altered. And it is exactly what we found. So either in MLPR mice or in lupus patients, after the treatment with the peptide, the level of the MHC molecule, which is increased in lupus condition, is again decreased. And a scrambled peptide, a control peptide, which is related to P140, but not exactly the P140, has no activity in this system. Okay, so now if, with my green arrow here, if there is a diminished expression of this molecule, what is behind, what is later in the process should be affected. And this is exactly what you have. T cells here are no longer able to react with other peptides, self-peptides, which we incubate in the cell culture. So this is in orange here. The cells, after treatment, cannot react with this, with the different peptides here. So we were, it was interesting to see the consequence of this, and the consequence is that here, all this production here is affected after treatment, so less activity of T cells, and therefore less activation of autoreactive B cells. This is what is seen here in mice, what is seen here in patients, and therefore we have less antibodies. And I remind you that these antibodies are toxic for the different organs. What you observe is that there is no direct effect of the peptide on B cells. We need this process attacking first the antigen-presenting cells. So, of course, we were very happy to show that, but we were afraid that the total immune system was affected. Our goal is to avoid this, because we want that the people treated with a peptide can react with and mount a response against a virus, a bacteria, whatever. And it is what we observe, what we did. We treated animals with the peptide, and then we infected these animals with the flu virus. And what we observe is that they were completely able to mount a reaction against the virus and recover normally after their infection. So clearly, this peptide is not an immunosuppressor, it is an immunoregulator. So here in brief, so the peptide enter the cells by the clathrin pathway, is present in lysosome, interact with HSPA8, and block all the process later in this model. So this is what I show here. So another view. So on the left, you have what's going on in lupus patients with, at the end, the attachment of autoantibodies in the kidney or in the skin, for example. When you treat with a peptide, you block at this level, and of course, nothing occurs after that. And recently, we developed a new mouse with a defective T cells, and we had exactly the same thing. So you will ask me, but why we target only the autoreactive response and not the response against the non-self? And in fact, it's due to our recent discovery of the model is that the P140 acts on autophagy. And autophagy is a process which eliminates the excess of self-elements, protein, misfolded proteins, or unstructured protein or proteins that are produced in excess. All the time, all of you are very active in autophagy. And in fact, in lupus, we observe that this process is hyperactivated. And the peptide is acting here in this process and block all the excess of autophagy found in lupus patient and lupus mice. So I will perhaps, so this is the marker we follow to demonstrate this. So Nicola was very active in this field and one of my first students involved in autophagy. And we discovered that more particularly the chaperone-mediated autophagy process was affected. And this was demonstrated in collaboration with Ana Maria Cuervo in New York. And we also in vivo recently demonstrated that we have a decrease of certain marker of CMA. Another student, Nicolas, showed that, in fact, the peptide has no effect on mitophagy, only on CMA in chaperone-mediated autophagy, showing that the peptide, by chance, it's impossible to create this tool, by chance, has a very specific focus on lysosomal autophagy, but not mitochondrial autophagy. So then we said, but okay, lupus is great, but perhaps we could use the same molecule to treat other autoimmune disease, close autoimmune disease, or distant. And we started to use the peptide in different experimental animal models. We started with neuropsychiatric lupus, and then with Sjögren's syndrome, with CIDP, with asthma, with inflammatory bowel diseases, gum disease, and so in all these models where autophagy is increased, we have a success treatment of the pathology. And today we have also a paper submitted in the field of gout. So, for example, only to illustrate by a small number of examples, here in the case of colitis, you know that in Europe, for example, there is 1.5 million people with ulcerative colitis and 1 million with Crohn's disease. And here it's very impressive because we use different models. And here, for example, we have the survival of mice when you treat with a negative peptide, and we have 100% survival with a peptide. And the body weight here increased again when the mice are treated with P140, while it is very low when the control peptide was used. And also we have effect on the colon length, which show the inflammation. And we use like that different model. We have less blood in the stool. The index of activity is again recovered, and so we had many improvement in this case. So this part of the work was done by a student coming from here, from India. Shruti was very effective. We had 2 very nice papers on IBDs, and she was really successful. So we tested other models, for example, Sjögren's model. So we had some also nice results in salivary glands, in CIDP, in peripheral nerves. And recently also we we had a program on periodontitis, which is really a dramatic inflammation of the gum. And here, just to illustrate, we had many, many results, but here something which really impressed our collaborators and ourselves is that we have a protection against the bone destruction. So this is very good because treating inflammation is okay, but if you can reconstitute the bone and to keep your teeth, this is of course very much appreciated. And now with asthma, we have also some very good results. And asthma is not an autoimmune disease. So here again, we are in a situation where you have inflammation, chronic inflammation, which is treated by the peptide. So altogether, and to finish, we have really a beneficial effect of this peptide in different situations. In all these models, we had an improvement of clinical markers of activity. I've shown bone preservation, a decrease of pro-inflammatory cytokines at the level of the gene, but also on the protein in target tissue, diminution of expression of biological markers, and inflammatory cell infiltration. We have less pro-inflammatory cells inciting nerves, for example, or lungs in asthma. And the marker for autophagy, which are expressed abnormally, are recovered after treatment. So you will tell me, but there are many molecules that can modify the autophagy process. Our molecule has the advantage to really to be here, to act here, and this is the in a very specific way, and not in different process like also apoptosis or necrosis or whatever. And we published a very nice paper with another Indian fellow who passed 1 or 2 years pre-PhD, and then several years again as postdoc in my lab. We have, I can tell you, 12 papers together for his stay in the lab. So Srini Bonham, you now spent 1 year in Paris and now is in the US, and we had a paper in Nature Review Drug Discovery with a very high impact factors. So this paper is quite important. So the strength of our strategy is that it's a mechanism-driven therapeutic mode of intervention. It is the unique mode of action that affect only the autoimmune cells but not the normal cells is also very important. And this explains why there is no effect, for example, for people that receive the peptide against the infection. And we use a very low dose of the peptide in contrast to what we have to inject when we treat with therapeutic immunoglobulins, for example. And this explains, again, another aspect, that we have a very few secondary effects, and the peptide is non-immunogenic in contrast to therapeutic immunoglobulins. And this is a chemically synthesized peptide you can produce by ton if you want. So, and with a very pure quality, so this is not a limitation of the process. So today what we are trying to do is to find a way to administrate the peptide in a non-invasive way, for example, oral, nasal, or transcutaneous, because today we administrate the peptide by the subcutaneous way and in animals by the intravenous way. We are also trying to develop a sort of diagnostic sheets to be able to differentiate responders versus non-responders, and this is for the future clinical trials, but also for the treatment in real life. So I'd like to finish by thanking all my students, postdoc and collaborator. On the left, you see that there were quite a number of the partners with me, and the current members of my lab, because all that is due to their work and their energy. And of course, you have our sponsor on the bottom of this slide, and CNRS, of course, and University of Strasbourg, but also other agencies. And I would like to thank you first to be here, and also the director of this institute, and also CEFIPRA and CNRS for making my travel and my stay here possible. Thank you very much for your attention.
Thank you, Professor. I think that was indeed very enjoyable by the audience. And if it's not too much trouble, there's been a request from the audience that would you be entertaining a few questions from them?
Yeah, sure.
Anybody from the audience, if you want to ask any questions, you can go ahead.
Very nice talk. There have been two important proteins called SMN and Tudor-SN, which have been also associated with some of these systemic diseases. Along with this peptide, because they might be working in a different way. So along with this peptide, can we also target those proteins along with this and then get a very more effective?
I don't know. First question, first problem. I have no answer about that. But it's true that we, in my mind, we are very lucky with this peptide called P140, but very probably there are others. We just now focus all our energy on this peptide, but why not another one? Or even several ones.
Extremely interesting talk. I just wanted to know what is the parent molecule from which P140 has been derived?
This is a protein, the U170K protein, which is a big protein present in the spliceosome. And the spliceosome is involved in the processing of RNA. And it is one of the protein recognized by the antibodies from patients with lupus, but also with mixed connective tissue disease.
Ma'am, I just want to ask that that autophagy being a very dynamic process and there are heterogeneity between different cells and organs, how this molecule specifically targets the T cells and the B cells autophagy. And another question is that...
I will answer with that because this question shows that you very well understood what, what I presented. I am very glad you asked this. And I have no response. It's what we are doing here. It's clear that with the amount of peptide we inject to individuals, it's a very small amount. It's impossible, just impossible that the peptide penetrate all cells, only certain cells. And this is my drama. I don't know what force the peptide to recognize certain cells and not all cells. And it's why we try to develop this diagnostic test to determine the amount of cells, the number of cells. If some patients have more cells able to to accept the peptide, this is a key question. Thank you for asking. Thank you.
So
Second question.
Another question that how it specifically distinguishes between chaperone-mediated autophagy and not other branches of autophagy, because all of culminate to some factors at some point of their pathway.
I'm not sure that I understood the question. I'm sorry. Okay. In fact, again, I don't know why, but this peptide interacts very well and very strongly with HSPA8. And we screen a number of, in the proteome study, we screen a number of other HSP protein, and this is only this one. So there is a motif in the HSP8 able to interact very well with the peptide, and this is the key. It doesn't react with other protein. And again, this is a protein which is always present, but the level of which is increased in case of inflammation. Thank you.
Hello, ma'am. Thank you for talk. Okay. First of all, thank you, ma'am, for such a wonderful talk. I just wanted to ask that, as you said that the peptide which you took P140, certain part of that peptide had immunogenic response, while certain didn't have. Because for that you did phosphorylation and other changes, alterations were done. So I just wanted to ask that how did you cross-check that the particular T-cell response was found against that particular part of antigen? Which technique was used to find that?
Yeah, there is several things. First of all, we had this peptide. When we screen, it was not phosphorylated. It was normal, you know. But we asked the question, why this, you have seen, everything is negative, only this peptide is positive. So we thought perhaps there is something, a motif again, in this peptide which render the peptide more antigenic, perhaps we had no idea. And at that time, it was said in the literature that post-translational modification are very, very important to create an autoepitope. So it's why we change, and because we are a lab of peptide chemistry, we had the facility to produce a lot of peptides, different peptides, and we change a peptide, acetylation, as I said, but ubiquitination, a lot of things. And the test we used was a cell culture test, so in which we incubate cells from patient or from mice with a peptide, and we measure the production of IL-2, for example, or IL-4, or whatever, and also the proliferation of cells by incorporation of tritiated thymidine.
Does the peptide remain fully intact in the lysosomes, or it is processed, little bit trimmed, and then it binds to the HSP?
This is also a key question. When we publish the results in the lysosome, the referees ask us how the peptide can resist with all the proteases you have within lysosome. Is it intact or not intact? So we did a lot of studies of stability, and in fact, we discovered that peptide remain intact with phosphorylation, but intact several hours, something like, would say, 24 hours, intact. And so we tested several possibilities. The non-phosphorylated peptide is destroyed in, less than 1 minute, and the phosphorylated peptide is very stable. And we think that, in fact, the phosphorylation hampers the degradation by proteases, by sterical entrance or something like that. So, in fact, it is protected by the phosphorylation.
Very nice talk, Dr. Sylviane. Have you tried the effect of this peptide on RA? Because that I saw
Which
that
kind of RA?
Rheumatoid.
In rheumatoid arthritis, no.
Particular reason?
No, I have several times the question. I think my problem is that there are several models for rheumatoid arthritis, and I don't know which one is the best. Because each model has a difficult aspect, and also this is one aspect, but I could solve perhaps. But I like to also to have a panel of very different diseases. But I know that rheumatoid arthritis or close disease would be very important in terms of strategy for clinical trials in the future, or something like that. Yeah. So I think I will do it. I will do it.
Hello, ma'am. It was a wonderful talk. Ma'am, I wanted to ask, like you mentioned in the clinical trials, you found that the peptide worked for the European population and in Mauritius, but it did not give a positive result in the US. So what could be the possibilities, and are you like working on that aspect?
I can tell you that it was really a shock when we learned the results, because you have seen that it's quite different in the treated population and placebo in the other countries. And in Europe or in Mauritius, the population is really different. And in the past, we had also some results in South America. So really, we have the feeling that it should work everywhere, and it's not depending on the genetic of patients, etc., etc. And when we had the result that there was absolutely no difference at all, between in the US, so we were extremely disappointed also to understand what's going on. And so we still don't know. It's why we repeat. So we think that probably the selection of patients was involved. We cannot say that it's a genetic background. We cannot believe in that. But probably the selection, you know, we are a small company. We are not a very big company. And I think that perhaps the selection of patients was carefully done for I don't know, big pharma, and for a small company in France. It was, I don't know. It's difficult to know, and it's why now we put again a lot of money and a lot of time also, because it was quite a delay to accumulate enough money to start again a phase 3. But we really believe, and we follow carefully the patient now that who will be involved in the story to have a successful phase 3. But yeah, it's difficult to know the reality, because we have chart and we have a lot of criteria for inclusion, and I don't know. It's difficult to know. Thank you for your question. Yeah.
If you add limiting amounts of your peptide to a mixture of autoreactive and non-autoreactive cells, is there any preferential targeting? Question one. And
Yes.
secondly, do you know why? So is there a receptor for it?
Yeah, it's now I have just a new PhD student who started just now in October. We know that the peptide enter via the clathrin pathway. But what I would like to know is the composition of this complex, because clearly the peptide should interact with HSPA8, but not only, with something else really at the entry complex. And we call this program PepDoor, because we have to identify the partner of the door of entry. And as you say, it is possible that either patients with inflammation have one of these compounds expressed in a higher quantity, and in normal cells, because the peptide has no effect whatsoever in normal cells. So perhaps this protein, this key protein in the complex, is not present at the surface of normal cells. And so ask again in 2 years, and I hope I have the reason.
While people, we have a lot of time for questions. So for running around, I'll ask 2 questions. Yeah. So when you have phosphorylation in a cell, there's also always a stoichiometric issue. There is never 100% phosphorylation while it is being processed, and so on and so forth. So, so in a natural situation, when you identify the peptide, it could be phosphorylated versus unphosphorylated versions, and so on and so forth. So the results are likely to be mixed in the wake of your answer that phosphorylation may stabilize the peptide and prevent it from lysosomal degradation. So how do you counter that problem? And
yeah, I can answer 100% are phosphorylated, because we synthesize that is phosphorylation.
Yeah,
so we introduce the FMOC amino acid as a phosphorylated one.
That,
so
that
100%.
That is for the therapeutic for aspects of it.
In vivo.
Yeah, in vivo.
In vivo, so first of all, this was a question I asked to a postdoc from the Netherlands who joined the team a few years ago, because I had no certitude that the peptide exists phosphorylated in vivo. So your question is quite pertinent. And we made the discovery that, in fact, in normal cells, this sequence is not phosphorylated. And the phosphorylation occurs, so this was the first result. The second, only when cells enter in apoptosis. It is linked to the very first steps of apoptosis. And after this phosphorylation at position 140, all the other phospholytic sites in this protein are degraded. So, and to answer, only apoptotic cells, but is it 100% molecule? I don't know.
Fine. What is the target of this peptide? What is the target? I know it is blocking the...
So the target is HSP8 when this protein is present. And as I said, the other question, probably another protein at the entry, altogether a complex. We have to decipher.
That would make sense because a lot of these HSPs tend to have affinity more for the phosphorylated version of certain proteins than the others. More questions?
Is the NMR structure of the phosphorylated peptide or unphosphorylated? And what is the difference between the two states structurally?
In fact, we studied the NMR structure of the two peptides in solution without TFE, so without a molecule able to induce an alpha helix. So you have seen the structure I showed in the slide. The structure of the non-phosphorylated peptide is very similar. For me, as an immunologist, no, they are different. But for specialists in NMR, they said there is no big difference. So, but this short peptide, 21 residues in solution, it moves to, you know.
And the structure of the peptide resembles that part of the protein as well.
This was the discovery, because the X-ray structure of the protein appears several years later we started the program.
So I was actually wondering, the non-phosphorylated peptide which has very short half-life, is it because phosphorylation stabilizes it and the stable structure actually can act?
So, but in the protein, we think that, of course, this sequence is protected by the other, you know, the environment. So there is no cleavage. This is only when we have the peptide in the tube, you know, that it is unstable when it is non-phosphorylated and very stable when it is phosphorylated.
Hi, Sylviane. Excellent talk. Just a practical question. Have you considered any combination therapy with the peptide? I mean, either with other immunomodulators like rituximab or IVIG, intravenous immunoglobulin. Would that be beneficial or not?
So the molecule you just cite, we didn't test, but what we did was in mice, of course, we tested with immunosuppressive molecule. And in fact, this does not affect the response against the P140. But some immunosuppressors are so active, so aggressive, that you delete completely the T cell compartment in vivo. And of course, then the peptide P140 has no action whatsoever, because it has to find T cells, you know, to have an effect. So if you eliminate these cells by an immunosuppressor or a killer, it's finished. So it's why the patient, perhaps the question of the individuals in the clinical trial received too much immunosuppressive drugs, and when we injected the peptide, there is no target cells to treat.
Two short questions. Have you tried other lupus models, and have you tried subcutaneous versus IV? Because you do subcutaneous in the human, IV in the MRL mice. So these two.
So we tested only once, and a long time ago, I have to repeat, in another model which is well known for lupus, is a BW mice. At that time, it was not successful, but we did only one experiment, so this has to be refined and repeat again. And your second question was?
Subcutaneous versus IV in the MRL model.
So the problem of model of mice is that the skin is not at all similar to the human skin. So we had no success via the skin in mice, while we have success in patients. But in mice, we tested IP with success. It was essentially IP and IV. So a systemic way of administration.
And one last question, please. In all the other models that you presented, do you think down modulation of MHC is the only mechanism in all of those models, or something else is going on that we don't know?
Yeah, something else. My point of view, I'm just writing the paper now, is that I think that in all these models, because they are so different, so the clinical and the cause of the disease is very different. So I think that the peptide locate in organs, for example, spleen, or another organs, perhaps lymph nodes or whatever, and from there we have an effect outside. And we can call that abscopal effect, for example, like we use in cancer. I think it's a distance. So the first organ is probably a lymphoid organ, and then we stop the progression of or infiltration of cells, other pro-inflammatory cells and cytokine. So we have an effect at distance. Otherwise, it's difficult to explain. Even we have in the target organs, for example, the lungs or salivary glands, we have effects, real effects, but I think it's due to a blockage of stimulus. Yeah.
Quick last question. So, have you thought about replacing serine with threonine, and then phosphorylated form of this peptide can have similar, better, or no effect actually?
I don't remember. I'm sorry if we have threonine in the peptide, but we have two serine. So what we did, we have two serine, and so P1 for the serine 140, and by heart 136, and 36, yeah. But when we produce a peptide, this peptide was not so soluble. So we just eliminate, because if you don't start your whole story with a very soluble peptide, you will have a lot of problem later on. So we just forget.
Thank you very much, Sylviane. And Sylviane will be with us for the next couple of hours during lunch, so you're most welcome to interact with her and ask questions.
Thank you for your questions, and I would now request Dr. Debashish Mohanty and Dr. Pushkar Sharma to please join me on stage and felicitate Professor Muller.
Thank you very much. Thank you. Oh.
You have to come in the center. Yeah. falling apart. We're not done yet. Vikra has another gift for her.
Oh. Thank you. Thank you. I'm really lucky. I will
Thank you so
come
much,
back.
ma'am, for joining us. Yeah. I now request Dr. Debashish Mohanty to please felicitate our founding director, Dr. G.P. Talwar.
To come back to NII is like finding your own self. I'm very touched that a lot of traditions of this institute are being maintained, and that we are honoring also people who are doing good work in relevant areas. So very good of you to have come and have addressed. It's what would be very nice is to also hear something about the relevant progress during this year of what Institute has done. I think in future, if you could also have a small presentation on that, it will be very relevant. At least on one day of the year, we should recall and appreciate the work which the students and scholars and staff of this institute have done over the year. You know, 6th October is the Founders' Day. My own birthday is on 2nd October. And so it's a very special month for me also. Thank you very much for organizing this excellent program, and keep doing, and also add to it a bit of what the Institute has done over the year. Thank you.
Thank you, sir. I now request Dr. Rahul Pal to join me on stage and deliver the vote of thanks.
So my name is Rahul Pal and I work here at NII, and proposing the vote of thanks is a pleasant responsibility. I'd like to thank, of course, Professor Muller for accepting our invitation to be here today and for delivering such a wonderfully lucid lecture. The studies she's described truly encompass bench to bedside endeavors for some of the most intractable, hard to treat diseases, all supported, as you saw, by deep scientific rigor and novel mechanistic insight. It's been a pleasure and a rare privilege for us. Thank you. I'd also like to place on record appreciation to the Indo-French Center for Promotion of Advanced Research, CEFIPRA, its director, Professor Nitin Seth. Thanks also due to Dr. Srini Kaveri, Director, CNRS Office in India Embassy of France, for making this event possible. The NII administration, as well as our technical and engineering teams, capably led by our senior manager, Dr. Deepak Vashisht, as well as Ms. Chandresh Bhaktani and Anju Sarkar, deserve special mention for helping us put this event together, for their expertise and their experience. And finally, thanks are also due to our young graduate student, Zikra, for anchoring today's event with poise and grace. Thank you all for being here, and please join us for lunch. Please join us for lunch. Thank you.
Thank you so much for this exceptional day. I will always remember, always.
Long live India and France friendship.